In this video, we'll take a look at the second step of virus replication, which is known as penetration and uncoating. Penetration is simply the process of the animal virion entering the host cell. Uncoating is the release of the genetic information, the genome, from the virion within the host cell. As you can see here, the manner of penetration is going to depend upon the type of virus that you're considering. Enveloped viruses, those that contain an envelope, can enter via two methods, fusion or endocytosis. Since non-enveloped viruses do not have an envelope, they cannot fuse. Ultimately, non-enveloped viruses enter via endocytosis. Unlike the process of penetration when it came to the phages, the entire virion will enter the cell in the process of penetration. Remember, this differs because with the phage, only the genome is allowed to enter in most cases. Once that virion has entered the host cell, in the case of the animal viruses, that is when you'll see the genome, the nucleic acid, released from the capsid, and that process is referred to as uncoating. So what you have here on your screen are two different manners of penetration. Up top, we have penetration via fusion, and down below we have penetration via endocytosis. Remember, fusion is a characteristic exhibited only by enveloped viruses, whereas endocytosis can be used by both enveloped and also non-enveloped viruses. You can see initially here we have the process of attachment. The spikes located on the viral envelope are going to be used, and they'll bind to glycoproteins on the surface of the host cell. We talked about this in the previous video dealing with attachment or absorption. Once that virus is attached to the host cell, fusion can begin. In this case, you have to keep in mind what a viral envelope is. Remember, a viral envelope is essentially a lipid bilayer. So in this scenario with fusion, the viral envelope is going to fuse into the cell membrane of the targeted host cell. So as we transition here, you see the fusion of the viral envelope with the host cell membrane. And as the two fuse together, the virion is released into the interior of the cell. Notice that we end up with the envelope of the virion and the host cell membrane integrated with one another. That is what fusion is. The two literally fuse together. And then once we've released the nucleocapsid, the virion, into the cell, you can see that we're going to uncoat the viral genome in this process called uncoating. Next, we can take a look here of entry via endocytosis. When it comes to endocytosis, whether it's a naked or enveloped virus, it will attach to membrane proteins involved in the process of triggering endocytosis. So you see here viral spikes being used to attach to membrane proteins. Next, you'll see extensions of the cell called pseudopods begin to extend out and surround the viral envelope. And inside, you would have the nucleocapsid. As these extensions of the cell membrane, usually referred to as pseudopods, extend out and surround the enveloped or even what could be non-enveloped virus, you're going to eventually form a tiny little endocytic vesicle. As the two ends of the cell membrane pinch together, you will see now that that vesicle is going to be allowed to enter the host cell. In order to be released, the virion now fuses or attaches from the interior of the cell to the cell membrane, and you can see as the vesicle and the envelope in this case, remember for a naked virus it would not have an envelope, but in this case as the envelope and the vesicle fuse, this releases the virion into the interior of the cell, where ultimately the viral genome can be uncoated. Once that viral genome is uncoated, there are several different strategies utilized for synthesis. It ultimately will depend upon the type of viral genome that you're looking at, whether it's double-stranded or single-stranded, RNA or DNA. What you'll find, however, in most cases is that DNA viruses typically are going to replicate within the nucleus, and that RNA viruses typically replicate within the cytoplasm. We'll discuss more in regards to that when we begin to go and take a look at the process of synthesis.